Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good afternoon, everyone. My name is Dr. Robert Freemuth. I'm an associate professor of biomedical informatics and the business owner of the Genomics platform at Mayo Clinic. I would like to welcome you to the Center for Individualized Medicine Grand Rounds, or CIM. The CIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation and demonstrate how individualized medicine is being translated into practice to meet current and future patient needs. Today, we are highlighting the theme of an omics data platform, one of CIM's 4 strategic areas, and which describes the infrastructure and systems required to centralize genomic and multi-omic data into a single clinical repository, enabling omic data to be delivered to the point of care in support of individualized medicine. It is my pleasure to introduce today's presenter, Dr. Nephi Walton, the Associate Medical Director of Intermountain Precision Genomics. Dr. Walton currently leads the Heredogene Genomic Sequencing Return of Results program. He also serves as the Associate Medical Director of Intermountain Sequencing Laboratory. He is the current chair of the Genomics and Translational Bioinformatics Working Group in the American Medical Informatics Association. Nephi has given several presentations about his work to translate genomic data into general medical practice, work that he started while at Geisinger, and which he is continuing in his current role at Intermountain Healthcare. I've had the pleasure of knowing Nephi for many years. We have had spirited discussions about the gaps that need to be filled to effectively implement genomic medicine and the technologies that are needed both now and in the future, and he isn't afraid to ask provocative questions. It has been fascinating to discuss the similarities and differences in the genomic medicine implementations at our respective institutions. It will probably not surprise you to hear that he encountered many of the same challenges that we did at Mayo Clinic with our Write 1K, 10K, and Tapestry initiatives. And he offered to share with us some of the lessons he learned and how he is addressing those challenges going forward. Today, Dr. Walton will be speaking on creating a home for genomic data in the EHR. Genomic data has the potential to improve health by informing clinical decision-making and individualized clinical management. Vendors of EHR and lab software have only recently begun to support discrete genomic data, and the standards that are needed to exchange those data are still maturing. As a result, many challenges related to the adoption and implementation of genomics remain. Aside from the storage, normalization, and management of discrete genomic data, new clinical workflows are required to support the delivery and utilization of genomic data that is generated preemptively and at population scales. New technical solutions are needed, and those solutions must integrate seamlessly with existing clinical software systems, including EHRs. Throughout this talk, please use the Q&A feature in Zoom to submit your questions. We have planned time for discussion at the end of the session, and I will be monitoring the questions that are submitted throughout Nephi's talk. For those who have not yet claimed continuing education credit, a message will be sent out in the chat. Now, please join me in welcoming Dr. Walton. Nephi, the floor is yours.
Thank you, Bob. It's been great working with you over the years on these challenging this challenging topic that I'm very excited about. Let me share my screen and we'll get going right into the presentation here. So as Bob mentioned, my presentation is about integrating genomic data into the electronic health record and really looking at how we can deliver population health genomics in a sense. The objectives for this talk is to be able to demonstrate knowledge of the current state of genomic data in the electronic health record, describe the implementation challenges related to the integration of genomic data in the electronic health record, and to identify deficiencies in current standards that impede the integration of genomic data into the electronic health record. Whoops. I apologize. I have nothing to disclose. I am the site PI on a couple of on an NHGRI and NHBLI grant. So just as some background, as you know, with this group especially, genomic data has a potential to improve health through targeted clinical management. And the integration of genomic data into healthcare is something that's been considered a national priority and international priority for several large organizations. But only recently have EHR vendors allowed for the storage and access of discrete genomic data. And in doing so, this has opened up a lot more opportunity. But there are still a lot of challenges that we face in adoption and implementation of these technologies. So I've been a part of 2 population sequencing initiatives and really enjoyed working on both of them. In fact, as my former mentor, Mark Williams, said when I was at the meeting with him at ACMG last week, if you want to recruit Nephi, you don't talk about dollars, you talk about genomes. And so when it was an easy decision, to go to Geisinger and work with all the genomic data they have there. So with their MyCode program, this is a whole exome sequencing study. It's a collaboration with Regeneron, and currently they have more than 350,000 enrolled participants. And I don't know the current number, but the last I knew it was over 180,000 of those are sequenced. And they have the Epic EMR. I recently moved to Intermountain Healthcare, where I lead the Hereditary Return of Results program with Brent Hafen, who's the head of genetic counseling. And there we're doing whole genome sequencing as a collaboration with Decode and Amgen. We currently have over 100,000 patients that are enrolled. Our target is 500,000. We have the Cerner EMR, but we have just acquired another healthcare system that has Epic, and we intend to keep them on Epic, and it'll be interesting to look at how we deploy across both systems. And we currently have 20,000 patients that have been sequenced through this study. So I did a couple of pilots at these different institutions. The first was at Geisinger. So this was using Epic's genomic indicators. And what we did is we took genetic data from 1,200 patients from the eMERGE network. And 142 of those had pathogenic variants for CDC tier 1 conditions and hereditary hemochromatosis. Some of the tier 1 conditions included BRCA1 and BRCA2 breast cancer susceptibility, Lynch syndrome, and familial hypercholesterolemia. And you can see the respective number of patients for each of those conditions. Now, these patients were somewhat selected for conditions. You wouldn't expect to see 142 out of 1,200 patients to have these conditions. So we also looked at pharmacogenomics for all of those patients. And this was actually expanded to about 2,400 patients just after or as I was going to Intermountain. So for these conditions that were shown, we did construct clinical decision support and patient provider facing information for all the variants. And so what the clinical decision support that would appear for the tier 1 variants was really related to health maintenance. And so what we would do is you would schedule MRIs, for example, and that would go into their regular health maintenance guidelines. One thing that we quickly discovered, and you have to be careful in with this, is that what happened is for Lynch syndrome patients, we were putting in a schedule for colonoscopy, and there were already guidelines in there for it based on age. And so you would actually have 2 reminders for colonoscopies that with discordant dates. So that was something that was an interesting challenge to work around. And we'll talk a little bit more about the way that was approached later in the talk. For the pharmacogenomics, we had best practice alerts, which if you're using Epic, you should be familiar with these. So they're little pop-ups that give you an alert about your medication based on the genotype and will allow you to discontinue the order and order something else. And in some cases, actually give you the option to click a button and order a new medication. And so we implemented these. And these are demo slides where I actually can't show the actual ones from our EMR based on Epic's restrictions. We also implemented genomics through the patient portal, so a patient could go in and see what their genetic findings were, and then we had pertinent information about those genetic findings, and then links out to other resources and ways to contact providers if they needed to help manage their conditions. So we do have some pilot results that were interesting from this study. So health maintenance, as you know, is a little harder to track, but we did get some good data on the pharmacogenomic results. And we had, from our initial pilot, we had alerts fire on 94 patients. 55 patients had alerts fired on new prescriptions, and 40 out of 55 resulted in changes in care based on alerts. So for new prescriptions, the clinical decision support was pretty effective. The one thing we really don't know, and this is an area that needs a lot of study, is, how did that impact clinical outcomes? And that's an area that we're working on at Intermountain now to try and get a better handle on that. And then 39 patients had alerts fire on reorder of existing long-term medications. This is a medication the patient had been on for some time, and nobody made any changes based on those alerts. So it's really telling about the effectiveness in those cases. And in most of those cases, the patients had probably already been dose adjusted. And so, there's some question about how you might tailor alerts in relation to that. But that was just a really interesting finding, that there were absolutely no changes based on those alerts if they were already on the medication. So Intermountain, the initial implementation, and this is done primarily by Brett Hill and Tyler Haverly at Intermountain Healthcare. So they created a passive CDS system that had a direct interface to the laboratory PGX results. So we have a pharmacogenomics platform called RxMatch. When we did this pilot, then we had about 2,500 results. Currently, there's over 10,000 results in our system. And it was looking at 17 pharmacogenes. And essentially what it is it's a tab or screen they can go to that will show what medications the patient's on and how they are affected by the different pharmacogenomic genes. And then also the thing I thought they did that was pretty neat was they pulled up the conditions that the patient had based on ICD-10 codes, and then pulled up medications you would use for those conditions and showed the pharmacogenomic interactions to help with dosing. So the discouraging thing about this is that, well, first of all, without active decision support, actually tracking metrics on this system is somewhat difficult. But what we could see is we could see who is accessing the page, when, and sort of relate that to ordering of the test. And the thing that was discouraging here is that no one was really looking at pharmacogenomics. the only person that would look at it was the person who ordered the test, and they were only looking at it immediately after the results came back, but they weren't looking at it again when they would prescribe another medication. And part of this leads to one of the first challenges, which is that we really need a home for genomic data in the EHR. As we worked on some of these processes, we meet with different groups, and I think one of the most telling groups we've met with is primary care, and how they use this data, and how little time they have to actually think about it, and how you have to drive it to them right at the point of care in just the right way in order for them to use it effectively. And part of the challenge that you face in this is genomic data has not really ever been front and center in the EMR, and Epic has created a place for it, which I think is nice. But we need to have, providers aren't used to looking for that information. There are very few people that will actually look for a pharmacogenomic testing report before they order medication, which opens you up to the possibility of liability, right? Because if you have information about prescribing and you don't use it, and the patient has an adverse event, there's some liability there. And so getting providers used to seeking that information or putting it in front of them is something that is really important. But if you have a home for it, similar to imaging and labs, then it becomes more a part of patient care. And I think this becomes a lot more important as we use more and more genomic data in patient care. Another challenge we met this is still a challenge today, is the adoption of standards needed to pass genomic data from the laboratory into the EHR. There are standards out there, and Bob is very involved in the development of those standards, but getting labs to agree and adopt those, it has been challenging. And so I think one of the most challenging parts of doing our first implementation was trying to get that data through a standards interface into the EMR. And as many of you know, one of the big challenges with working with the EMR team is they handle all of the EMR tasks for the entire organization, and genomics isn't always on the top of their list in terms of priority. And so getting an interface built to bring that data in was challenging to the degree that I actually pulled the technical aspect out of it into my team, and then we built it and delivered it to them, which was much faster. I think we did it in a week, and the estimate we were given, I think, was 8 months. So that's not something that I know everybody can do, but that is something that's evolving. I think that has been addressed with several laboratories now. So things are a little better than what they were when I started in this, but it's not so much the adoption of standards as it is better translators for specific labs, which is really the way it has to be approached right now, unfortunately. The standardization of genetic phenotypes is a big deal. And I actually thought I had updated this slide, but when I was speaking to Bob about this presentation, he had mentioned, it'd be nice to see a representation of what this means. And so I spent 5 minutes doing a Google search in different standards, vocabularies, terminologies, and how they represent hereditary breast and ovarian cancer. And I found they were so highly discordant. In fact, the amazing thing to me, it's actually much worse than I thought it was. I thought, I'll find a few examples of how they're described differerly, but it was actually harder to find ones that describe them the same. And so you might have someone that'll say familial breast cancer, or hereditary breast and ovarian cancer, or hereditary breast and ovarian cancer BRCA1, or hereditary breast and ovarian cancer BRCA2. And there's some that will couple them together. If you look at OMIM, it's different than Mondo. And so there's not really a standard way to get this information. And this is really challenging because this is your point of decision support. And this is the area, this is where you're going to launch all of your maintenance guidelines and any of your pop-ups or reminders or things that come up. And so you're going to be getting these results from different laboratories, and they're all going to be calling it a different thing. So that's one of the big challenges that still remains. And right now it's just a matter of translating that information to a standard from specific laboratories, unfortunately. And when you try to look for a standard that covers this, there have been a few that have been suggested, but none of them actually are complete, I would say, for this right now. And then the other challenge is if you try to look at somatic genetic phenotypes for cancer, for example, or germline Mendelian conditions versus pharmacogenomics, those standards are going to be different. And so it's likely going to, I don't like to advocate for the creation of new standards, but perhaps it's modifying existing standards that cover those fields well, and then sort of putting something together from that perspective. But this is an area that needs a lot of work. And here, this went on the wrong slide. I apologize. This below here is the example of all the different names that I found on a 5-minute Google search. And these are things, this is from Omen, Mondo, Gene Reviews. I can't even remember the ones off the top of my head, but I looked at several common places you would look as sort of a reference of what this disease is. And they all have very different names. So the other challenge is the addressing the role of the LIS in the genomics process. And when we first did our first pilot, the biggest challenge was that our LIS actually didn't handle structured genomic data. And so we simply bypassed that and sent the data directly into the EMR in terms of the structured variants. And unfortunately, that's the process and path you have to take in some instances. At Intermountain with Cerner, it was different. We have our LIS did support structured data, and so we can actually bring that directly into the EMR like we did with the passive decision support. But that is a question, when you're approaching this, and the first question, of course, is does your laboratory information system actually handle structured data? As many of you know, this field changes rapidly. And so one of the big challenges is the maintenance of clinical decision support. And you really have to have a team that's dedicated to keeping up with guidelines, and teams that are involved in you'll have to have specialty teams in multiple specialties that can keep up on these things, because they do change rapidly. And there aren't any really good resources out there that manage all of that right now, which is my next challenge, the lack of standard information resources. There are a lot of different information resources out there, but they don't have standard interfaces to access them, such as info buttons. And they don't have a standard format or way to deliver, and they don't all encompass all the information that you would need. So this is another area that needs a lot of work, especially when you're an institution that might not have the internal resources to build what's needed here. One thing that was really interesting is the perception of discrete genomic data versus scanned PDF. So scanned PDFs seem to have this mystical power where they can remain hidden from providers who might misuse them, or that's the perception. And in reality, there is some truth to that, in that people, primary care providers aren't necessarily seeking out those things in a lot of cases, and won't necessarily know how to interpret them. And there's a lot of fear about taking what's on that report and putting it front and center on the problem list. That gives it a lot more power in a sense. And there is fear about, there's more fear about misuse once you put structured data and put it right on the, in an area that's similar to a problem list, in a sense, a genomic problem list. And so that was something that it was, I would say we do have to be more careful about what goes there, because there is that possibility. But I've seen PDFs that have been misused too, where variants of unknown significance have been acted on when they really shouldn't have, because they didn't have any meaning and were taken out of context. This is a big problem, and this example I have at the top is something that is actually the case right now. So variant classification and maintenance across patients and reports is extremely challenging, especially when you introduce structured data. So you can actually reclassify those variants based on a patient's clinical presentation. Now, as physicians, we commonly do that, but we usually talk about it in our notes. We don't necessarily, change anything on the report. But what that can mean is that you have discordant classification across patients and reports. So we have an instance where we have 3 different patients with reports from 3 different labs that have, one of them has a different interpretation. And in this particular case, there are pretty drastic implications based on this variant. And so you can see how this could potentially lead to a liability if one of these people are wrong and you act on, you act on it in a way. So for example, someone gets a mastectomy and they really shouldn't have because the other classification is right. And you have in your system that it's right from one of the labs, but not from the other. When you put it there in structured data, I think there's more of a fear of why are you classifying variants discordantly across patients? And you can actually look at that really quickly with structured data, especially if you're tracking information around those variants, which is another question. So if I'm a physician and I feel that there's a strong indication to reclassify one of these variants, should I go through some process to reclassify all of them and recontact patients that were classified a different way? So this is a big challenge, and I don't know that we have really good answers for that. And there definitely, from an informatics perspective, with existing systems, isn't anything that handles that well. The next challenge is the disparate needs of primary care versus clinical genetics. And really early on in our first implementation, it was easy to see that the same system and workflows are not going to work for one versus the other. And we actually totally abandoned the clinical genomics workflow because that affected a much smaller part of the healthcare system. And the needs were completely different. Primary care doesn't want to see variants of unknown significance, but those are very important to clinical geneticists. The way they use genetic data is entirely different. So that's something that's really important to take into account as you approach these. Interestingly, Cerner and Epic took different approaches, and I think it's been a good learning experience for me. Cerner, as they've started to build out some things in genomics, they started building from a clinical genetics perspective, whereas Epic had approached it more from a population health genetics view, which actually, and I don't know if that was intentional or not, because I think they had the idea when we did our first implementation that it worked for both, but it was clear that there were challenges in meeting the needs of both of those groups with the same product. So I've talked to a lot of people about this. So since I did our first implementation at Geisinger, a number of people had contacted me and asked, with all these challenges that you've run into, should we even start? And that's a really good question. And actually my answer is different depending on the organization and the resources they have. And the answer to that question is also evolving. Because some of these solutions are getting better. But the challenge you have right now is, I think that it is worth using the home that's been created in native EHRs for genomic data. But I think to use it, you really have to have a team and the support to make it work. And so if you don't have the resources in terms of specialists and genetic counselors and et cetera, to really build this out, it's not something you're going to be able to do really easily with the current state. So I think large organizations that might have these resources, such as Mayo, I think that this is a good idea to pursue this. If you're a small organization that has very limited specialist resources, not a lot of genetic counselors and geneticists, I think it becomes a lot harder. That said, I'm really trying to take geneticists out of this equation. At Intermountain, there are only 2 of us. And I interact with the patients from the population health level infrequently, actually not at all to this point, because it's handled primarily by other specialists and genetic counselors. And that's really where it needs to go, because we're not going to scale clinical geneticists. So the question I ask every morning now is, I have 20,000 whole genome sequences. How do I use this information to impact patient care? And I love being in this position, because there are so many exciting things that we can do with genomic data that can have a positive impact on patients. So I want to talk about an approach now that we've talked to some of the challenges. at least from my experience in how we approach this problem from an IT and IT infrastructure perspective. So one thing that I want to make clear is clinical genetics is not similar at all to population health genetics. And if you think about this from a clinical genetics perspective, I think you're going to miss the boat and you run into a lot of challenges. And from the clinical genetics perspective, I was at ACMG and they did a hackathon that was great. And there's a lot of fear about how you translate this into the front lines in terms of secondary findings and variants of unknown significance. And I think that the fear, I would say I don't have the same fear because we've done it at 2 institutions. And if you do it right, then I think it's fairly well received on the front lines. Of course, it's very important to include the people on the front lines in your processes. And so that leads into my next things. We're not as special as we think. I think that as clinical geneticists, as a profession, and I'm a clinical geneticist, so I can say this, I think that we've always thought that genetic data sort of had this special power and that it is beyond the comprehension of primary care physicians and other specialists. And I think that we have to lose that because right now, wait times are high for clinical genomics. There's not enough providers in the pipeline to really scale to what we need. And the genomics data and what we can do with it is growing just exponentially. And so we have to really think about how we can deliver it to other specialties and to primary care. And I would say the same thing is the case with primary or with specialists. So one thing I've encountered with every time that we're going to return a particular disease or gene, then we put together a committee that looks over, what we need to do with the patient, develop patient information and physician information. And the thing that I always have to really push on is to say, we need to give something to primary care providers so they feel comfortable managing this condition and feel that they're a part of the patient's care. And I universally get pushback that's, oh, you know what, just refer them to our specialty clinic. And I have to push to say, the person who manages this patient's overall health really needs to know what's going on. They're the one that's going to see them the most frequently, and they can put them back on track if they get off. There's also this fear of genomic data, and this helps if you really show them, in a simple way what this really means and what their role in it is. Then it makes it a lot less scary, and actually, and from my experience, it makes it exciting for them, and they're actually very engaged and very interested. So we need to lose the thought that we're so special that no one else can handle some of the things that we do. There's some of it they can. And this leads into, we need to dispel the fear of genomic information. This is present, I think, with pharmacogenomics. It's present with your tier 1 conditions, Mendelian disease. It's something, as I had talked to some of the primary care providers at Geisinger and the same at Intermountain too, there's sort of this, man, I wonder if I get one of these mycode or hereditary patients, what am I going to do? I've never treated these things. But if you deliver that information with the right information in the right way, then again, it's not something that's scary. And once they start getting it, they're more excited about it and excited about the impact it can have on their patients. For the most part, doctors, they go into this field because they want to help people. And if you are giving them information, then you can demonstrate, that what that's going to do for them. There's a really positive response. And so we need to work to dispel the fear of genomic information. Variants of unknown significance are actually insignificant. And this is the case for the initiatives that I've worked on because we're screening healthy people, right? So there's not that pretest probability of disease. And so variants of unknown significance really don't have a strong role in what we do, and we would never return one. This is particularly important when you talk about conditions in oncology where they might take drastic actions such as a mastectomy or a BSO. These are things that you have to be really careful and conservative. So from a population health perspective, variants of unknown significance don't have a role clinically What we do is reassess them over time and then, put them back into the pipeline once we know more, because you have to be very careful about, it's sort of this balance of trying to make sure you're helping everybody you can, but not going so far as you could actually do people harm. Another thing that is really important is to have realistic expectations of the frontline providers. When we've had meetings with this group, they are excited, they want to do this, but they have these 10 to 15-minute encounters where they have to do a lot of things for patients. And you have to give them information in a way that they can digest it relatively quickly, right at the point of care, because the how fast they operate in clinic. And so when you ask them, so hereditary hemochromatosis is a really good example of that, right? So that's when we've done a lot of results on. And that doesn't go into a specialty pipeline until, your ferritin levels get really high. And they manage that, but just giving them something that's really simple that explains it, that tells them what they need to do, what their role is, and when they need to refer, you can distill that down to something that can be digested in a minute, if you do it right. And so that's something that we've worked on with these conditions, is to make sure that we can deliver it in a way that they can use it right at the point of care, really quickly and effectively. Make sure you include a patient voice. This is something that's really important. We're doing all this for patients, and we need to make sure that we're delivering care in a way that patients can digest and use it, and especially with the way things are changing with technology, and also realizing that some patients won't have technology. And so you have to have several modes of delivering information, and you're going to have people that will digest it in different ways at different levels. So as you're approaching this, you have to think about that. Chatbot's a great thing, but there's a lot of people that are not going to interact with that or even have the means to do that. I think it has a place, and I think there's places where it's not good to use it. And then, just talking to patients about the way they would like to receive information and what they want to know. This is something I can't overstate the importance of. The other is to utilize IT to increase efficiency of the genetics team. The genetics team is obviously very important in this, although I've talked about minimizing the interaction and sort of relying more on other providers in the system. I think that that's important, and one of the ways that we can maximize, particularly if you have small teams, is to use IT to help deliver information, to help communicate things to patients. So that's something that we've put a heavy emphasis on. But again, making sure that there are ways to help patients that might not have the resources in terms of a smartphone or a computer, or have the knowledge of how to operate some of those things, make sure that they're not left out. So Bob asked me a few pointed questions. So I wanted to give some feedback on some things from my perspective. And of course, I'm not the know-all end all of all this. There's probably a lot of different opinions, but I'll just give mine based on my experience. So one is to use native EHR functionality versus a third-party solution. In my opinion on this is that you use the EHR for what the EHR does well. I've pushed really hard on Cerner, and I think I'm making some good progress based on their last response to get a home for genomic data there. And the reason why is because they have a whole infrastructure that's built particularly for decision support around that is Cerner, and you won't, with a third-party solution, you're not going to have as seamless of workflows and seamless integration. And so I feel like it's really important to use native EHR functionality, because it makes the workflow more seamless. And to my next point, if a home has been built, live in it, right? So if they build a home for genomic data, I think that's a great place to start, and it encourages the adoption, and it creates a place where physicians and other clinicians know to go for that information. So I think it's important to use that where it's applicable. But what I would say, I wanted to add a little bit more here. There are some things that the EHR will probably never do well for genomics. And so I think there are third-party applications or home-built applications that are important. And we've sort of taken that hybrid approach. But as we've assessed third-party applications, the real thing is to determine the value that it actually adds. And when you account for costs, think about the cost of integration and maintenance. Sometimes that's actually going to take more time and more resources to integrate than it will to actually build out some of the rules. So my approach to third parties, particularly for CDS rules, was that from my experience, most third-party CDS rules are not going to be adopted. Most of them will need to be modified. All the CDS that you're going to put in there has to pass through committees for adoption. And so you start to question, what's the value? If there's actual CDS capability within the native EHR that we can utilize, are we better off just building and maintaining that? And that's the thought process I went through. What is the value they're actually adding? Because with the time that we have to put into modification and adoption and all these things, it's really not giving us a lot of benefit. And so, as I mentioned, I think the ideal model is a hybrid solution. So we've done a lot of our own work to build out some of these things. And then we utilize the native EHR in places where it is most appropriate. So I think that, if I was to give one recommendation, that would be the recommendation I would give. And sorry, I had a little delay there. The other thing is, I know the challenge that a lot of places face, and this is pretty universal, I'm sure it's the same there, is access to the resources that maintain the EHR and build out some of those clinical decision supports. So one of the ways around that is really to adopt existing processes and infrastructure. So for example, with pharmacogenomics, there's already a group at most institutions that will manage, drug-drug interactions, et cetera. And so utilizing that existing process so that you're not waiting, so that you have a seamless way to keep adding and modifying, just adding somebody that kind of works with that group to put those in there, and looking for ways to do that so that you're utilizing existing workflows, so you don't have to request additional resources, if that makes sense. So try to utilize existing pipelines. And I think that's a key to success in this area, because you can get a lot of support maybe, after you lobby for, this is high priority, it's a good priority for our institution, you build it out, but then what happens with maintenance down the road? So that's something that's really important as you approach this. Build versus buy in terms of some of the ancillary things, frankly, there's not a lot of software out there that can handle particularly return of results programs. And so that's something that we've done a lot of building because there wasn't software to buy, actually. So that's something to consider in this space. Another approach, or something that Bob had asked me about, is genomic storage and access, and whether to use EHR vendor solutions. The EHR vendor solutions I've seen to date, and again, they've probably advanced past what I'd seen before, were not quite adequate. There were a number of concerns, and this is something that I would say with all third-party software, is a lot of times you get PhDs that develop things and don't really have a lot of clinical insight into how they're going to be used. And that's sort of what I've seen to a large degree. And so just be careful there, and then realize that what you adopt, it's going to be fairly dynamic because it's a rapidly changing field. And so for our purposes, it made sense to build because this is dynamic. The current things that were offered didn't fit all of our needs. There were some glaring problems with some, and we need this system to deliver genomics, in our particular case, both to research and to clinical care. And so having something that could serve both needs is important to us. So we ended up just building our own solution there. IT is critical to scale. When you launch these programs, you're going to be impacting a lot of patients. It's going to be at least 3%, but some of our latest data with the new ACMG list is actually looking like it might be significantly higher, especially if you look at targeted populations. When we look at patients, for example, that go into cardiology clinics, our initial data is showing that you're going to have 40% more variants than you would just in the general population. And so that's a lot of people, and you have to have IT infrastructures to support that, or it becomes kind of a disorganized disaster, and you lose track of a lot of patients that really should be receiving precision care. And to get that IT support, you have to have support from leadership. I mean, that's critical. Another thing to keep in mind with what we've done, this is starting from a research cohort, which introduces a lot of additional challenges and changes the approach. So if you were, for example, doing targeted sequencing based on people who met criteria for certain cancer syndromes, that's very different than population health, because there is a prior probability, and you might want to consider variants of unknown significance more. But I would say if you're doing, truly a healthy population, you don't even want to consider those. They shouldn't be in your equation at all. And you would just watch for changes in classification over time for those. So this is a simplified diagram of our return results process. It's actually, the technical infrastructure behind this is actually extremely complex. And there are a lot of moving pieces, and because there hasn't been a lot out there, we have had to build our own. This is a very simplified diagram of kind of illustrates what we have to go through. So we have our sequencing, our variant filtering, we have a variant review committee, we recontact the patient, we do a CLIA confirmation, we have to send them to genetic counseling, do cascade testing, and then all the clinical management pathways. Now you can imagine the work and the technical architecture that's required to manage that entire workflow. It's actually quite complicated. That's something to think about as you approach these problems, is what really building a good technical infrastructure underneath it to deliver care to the front lines. Two keys to success in implementation, I just always end on these because I think it's really important, is that support from leadership to prioritize the informatics component of this is extremely important. I can't overemphasize, I mean, I'm an informaticist, so that I'm a little biased, but that part is absolutely critical. And then the other is to realize that IT is only a small part of the framework required to make this for a successful deployment. You have to have an infrastructure to support this, a place for physicians and patients to go when they have questions. There are a lot of things that need to be in place to really make this successful within specialty groups and primary care and in your central leadership. So something to realize as you take these problems on. So just wanted to acknowledge a team at Geisinger and Intermountain that did a lot of this work, and I will take questions.
Thank you very much, Nephi. I really appreciate the talk. You've touched on so many things that we've experienced here at Mayo Clinic as well. And I know if we were all in person, and the audience would be head nodding as they applaud. So thank you very much for your talk here this morning. I would like to remind everyone, if you would like to ask a question for us to consider as part of the moderated discussion period here, please go ahead and put that in the Q&A feature down at the bottom of your screen. Nephi, there are so many things here that I wanted to touch on, and I know we won't get to all of them. So I'm going to try to hopscotch around to a couple of different topics, and maybe we can address a few things simultaneously. You mentioned, and I think I'm going to start at the end of the pipeline where we touch the practice, and then maybe move backwards. In your introductory slides, you were talking about the pilot that you did at Intermountain with Pharmacogenomics, and you had some statistics there about the number of alerts that fired and what kind of impact that had on patient care. That was at Geisinger. I was struck when you were talking about Intermountain that you indicated that a lot of providers never actually went back to look at those results in the future. Now, that's something, of course, that we've seen as well. I'm wondering, as somebody that is a little bit closer to the clinic and has thought an awful lot about this, How do we address something like that? Is this really pointing at a system that's needed to deliver information to providers without requiring them to actively search for it? Or what other thoughts do you have there?
Yeah, so I think we've actually just had some meetings on this recently, because that's a huge problem. And what we're talking about is ways to alert providers to things without interrupting their workflow. And there are ways to do that. Part of the challenge we're facing is where does that go? What does that look like? And that's part of what we're working on designing right now, because they want to know. Providers want to know if there's something, and this is something they're saying, I want to know this, but I have 15 minutes and I have to do all these things. How can you get it to me without making it 17 minutes, right? And so that's something we're working on. And I think, the answer is really to put it in their face, but not interrupt their flow, if that makes sense.
Is that capability that exists within the EHR today? I know you showed some screenshots there of some things that you were experimenting with, or are these new technologies that don't exist commercially yet?
So I have to say that I'm not certain with Epic, because I haven't worked with Epic for a while on this since I've been at Intermountain. So but that wasn't something that was presented as an option when I was working with Epic on this. I can envision ways that it could be possible. It is something that I'm working with Cerner on. I don't know exactly what it's going to look like, to be frank. We had touched on it with Epic. I don't know that we landed on a solution and had decided to not pursue that at the time. They are moving forward to expand some of the work that I did there, so they may approach that differently as too. And as I mentioned, we've purchased a healthcare system that also has Epic, so we're gonna approach it. But I haven't done a lot of work in that area yet.
There was a question in the chat that asked about genome level scales. When you have all of a patient's genome, how do you make a decision about where to store that? Is that something that you think about putting into the EMR, or is it in a different system? And I had a question related to that as you were talking about the tension between the capabilities of laboratory information management system and the EHR itself, and thinking about the different roadmaps and priorities and development cycle that each of those products have. And as an institution, thinking about the capabilities that exist now versus in the future, how do you make decisions as to where to store the data, knowing that all of this is essentially a big moving target?
Yeah, so one of the advantages that I have at Intermountain is that we actually have a sequencing laboratory in-house. And so, and I'm also, associate medical director of the laboratory. So it makes it, having both the clinical role and the laboratory role makes it easy for me, easier. I shouldn't say easy, but it's easier to control that with genomic data. Now, the reality with most institutions is there's a lot of different laboratories they're getting this information from, and they're not always in-house. And so the decisions that I made might not apply to other places, but the question you have to ask is, where is the clinical actionability decision? Where are those going to be made, right? And who's going to make them? And as a clinician, I like to think that we're better at making those determinations than laboratories, but laboratories are the ones that give us that information that we could base those decisions on. And so I think, in the long term, there needs to be better collaboration between laboratories and the clinical side. And this is evident, I think, in our ClinVar database and variant classification. There needs to be feedback. If physicians reclassifying something, they should send that back to the laboratory with the reasons. So the laboratory has that information and it goes into ClinVar the right way. Right now, people are looking at the same patient, the same variant, and they're sending different classifications into ClinVar, which is kind of crazy and frustrating. So it's easier for me. That's a hard question to answer when I'm not in the position that I'm in. In the position that I'm in, they're kind of, they kind of flow together. And so it was easier to say, you know what, we're going to just put this data from the laboratory in this database that both the laboratory and the clinical side use, but the clinical determination is, in this case, is actually a combination between clinical teams and laboratory teams. And how you build that outside of an integrated system that has that laboratory is something that needs to be addressed, but I think that's the most efficient way.
You know, on this topic of classification, you alluded to this just now in your response. There was a couple of questions in the chat about this as well. Wondering if you could comment a little bit more about the role that the laboratory and the provider might play in this process of understanding a variant's potential impact in present day and then the reinterpretation of that variant over time. How do we deal with things like non-concordant variants or interpretations that might be submitted by different sources? And what is our clinical and technological requirement around managing all of that?
So I'll say one thing that's positive first is that our knowledge of genomic data and what variation means is increasing every day. And so over time, that won't be as big of a problem. Right now, it's a big problem. And I think that we have to be cautious. And there's this balance of, so if I'm returning a gene for familial hypercholesterolemia, the intervention is not, first of all, you can clinically correlate it to see if there's an intervention needed. So, and then the intervention is not really drastic. If I'm returning a variant for BRCA and someone's going to get a mastectomy, my levels of conservative, how conservative I am change, because you're trying to find that balance of what's going to be the best impact on the patient. So one of the advantages we have with the population health is that we don't actually get a CLIA report until we have validated that we think the variant is pathogenic through a couple of different pathways. And so we don't have to discuss variants of unknown significance to patients because they'll never receive them. And this brings up a bigger question of where geneticists need variants of unknown significance and primary care physicians don't. Should there be some kind of system where you can order a lab report and you can request not to receive those until they're clinically actionable and get them later? And I frankly think that's something that needs to happen because they just introduce problems and potential serious implications.
I think we are very close to the end of the hour here. I've got a couple more questions, and due to time, I think I'm going to try to combine them into one. So apologies if this feels a little bit convoluted, but I think they kind of get at the same thing. One of the topics that you discussed during your presentation here was related to the different needs that consumers of these data have. You made a specific example about a primary care provider versus a clinical geneticist, and that they would need different information presented to them. There's a scalability issue there, I think, with respect to how do we build systems that at the one time, or on the one hand, meet everyone's individual needs and preference for consumption, but don't require us to build one-off solutions for every subspecialty that might exist out there. You also, I'm going to tie this into the second piece then, which is use the EHR for what it does well. And you talked a lot about how, especially for decision support, automated decision support, that's a great way of delivering that information. But then you flipped and said, we built a lot. And I'm wondering if you can comment on the other side of that coin about how do you know when to use what the EHR has and when you decide when to build, and how can we tailor this to each of the consumers?
Okay, so what you build, I think, will depend a lot on your resource. It'll depend a lot on the resources, and then just thinking about what EMRs do well and what they don't, and what you think they can do, they will do well in the future. And part of it is what kind of control you have over how that is going to develop over time. And if that level of control, lack of control that you have is going to limit what you're able to do, I think that's when you have to start looking at building your own infrastructure. In terms of the spectrum of what you would have to deliver for a gradient of information, in a sense. That's something I've thought a lot about because, you can say, oh, here's a special way to look at genetic data for clinical geneticists, but there are a lot of neurologists and cardiologists that specialize in genomics that will want that level of detail. And so as I've thought about it, I've kind of thought about, and I haven't solved this problem, but I've thought about separating into two tiers. And so you have a tier for people who think they understand genomics, and a tier for people who really don't have time to know about it and really just want to know what's this disease and what am I supposed to do. I think the challenge there is that how do you determine who fits in each tier? Are you going to insult people in the process? And is there a danger in opening up structured data to some people who don't understand it? And I've seen bad things happen, like people get scheduled for C-sections for Lois Dietz for a variant of unknown significance that was completely meaningless. That's a hard, I think you have to do something. You can't, if you worry too much about all the little challenges, they're going to be there. You're never going to solve everything, but try to get 99% or 90% right. And that's sort of the approach I'm taking.
That's great. Thank you, Nephi. I think the lesson here is there's a lot of work yet to be done. There's tremendous potential in this space, and don't let the perfect be the enemy of the good.
Yes.
So thank you very much for presenting at Grand Rounds today. I really appreciate you sharing some of your thoughts with us. Thank you to everyone who attended this afternoon. If you could, please take a moment before logging off to fill out the survey that will appear at the end of the session. Have a great afternoon. Thanks, everyone.